Hello dear students. So in my last class I was explaining about the theories of precipitation. So precipitation theory, it involved two different types or two different physical process. So they are, one is nucleation and crystal growth. So I explained about what is meant by nucleation. So that is the formation of the smallest particle of a precipitate capable of spontaneous growth. So that is what is called as nucleation, and also I explained about crystal growth. Further, so we understood what is meant by co-precipitation. So in that, solid solution co-precipitation and adsorption and occlusion co-precipitation was discussed. And next, so we studied about post precipitation. What is meant by post precipitation? That is, it is the precipitation of an impurity. So after the main precipitate has been formed. So the post precipitation also introduces appreciable error. So this was what is meant by post precipitation. So further we saw what are the differences between co-precipitation and post precipitation, and advantages of gravimetric methods were discussed and disadvantages of gravimetric methods were learnt. Okay. So this is where we stopped in our previous class, and in this session we are supposed to start with instrumental methods of analysis. So what is meant by instrumental methods of analysis? So till now, so we are studying methods of analysis. Okay. So what are the methods of analysis which we are going to employ in analytical chemistry as well as in other branches of chemistry? So chemical analysis means determination of composition of a chemical compound in a known or a given sample or a solution is known as a chemical analysis, right. So there we saw qualitative and quantitative analysis. So these are the two different classifications of chemical analysis. So these two, qualitative and quantitative analysis, are carried out in our hands. Okay. So we are not using any instruments to do this qualitative or quantitative methods of analysis, whereas so we are going to employ or we are going to use certain instruments for the analysis, for the chemical analysis. So what and all the instruments we are going to use, and the methods of analysis which we will perform with the help of instruments. So they are called as instrumental methods of analysis. So in this method, the quantity of substance is estimated using some properties of the substance. Okay. So what are the properties? So they might be the optical density or the absorption. Okay. So optical density of that particular solution, or we are going to measure the absorption. So how efficiently the solution or the compounds which are present in that solution, they can absorb a radiant energy or a light energy, or the percentage transmission. So these are all the physical properties. So even we are going to study the percentage transmission. That means so how much capable does that particular solution is to pass through the light from it. Okay. So when a beam of light or when a ray of light falls upon the substance, so how much of capability it has to allow the passage of that particular light. So that is measured in terms of percentage transmission, and also the electrical properties. That means this is particularly studied in electrochemistry, that is how much of electricity it can carry with it. So in terms of that we are going to calculate the conductance, we call it as conductance. Okay. So that means it is the reciprocal of resistance, that means how much of electricity does that particular solution will allow to carry along with it. Okay. And intensity of the color is measured and also the electromagnetic radiations. So, at what wavelength the excitation of the electrons are going to take place. So, that excitation and emission of the electrons from the lower energy state to the higher energy state. So, in terms of that, so we will measure the electromagnetic wavelength. So, all these are different physical properties which helps us to estimate the quantity of the substance. Quantity of the substance means how much amount of substance has been present. So, what is its concentration. So, how much in terms of mass that particular substance is present. So, all these will be measured, that is, so the quantity of substance is estimated using the physical properties, the physical or the chemical properties of that particular substance, like optical, electrical, intensity of the color and electromagnetic radiations, that is electromagnetic waves. So, all are directly proportional to the quantity of the substance. Okay. So, that means so how much of optical density is observed. So, it is directly proportional to the quantity, how much mass of the substance is present. Suppose if the mass of the substance is very less, then obviously the optical density also will be very less. Okay. In other way, so if the intensity of color is also directly proportional to the quantity of the substance. Suppose if we take more grams of compound, then the intensity of color also will be more. Okay. So, for example, if we prepare the copper sulphate solution, so copper sulphate is blue in color. Okay. So, suppose if we take 0.1 gram of copper sulphate solution, then the color the blue color which is going to appear, so will be of light blue color. Suppose if we take 1 gram or 2 gram or 3 gram, then the intensity gradually increases. That means, so the quantity of the substance is directly proportional to the intensity of color. Okay. So, these properties are measured at various concentrations using some instruments like spectrophotometry, conductometry, potentiometry, etc. Okay. So, the different techniques available are spectrophotometry. Okay. So, spectrophotometry is the instrument, so which is going to measure the color. Okay. Spectrophotometry. Okay. So, here spectra means color, photo means light, metry means to write. Okay. So, we are going to write the color of the solution or the color of the substance, that is intensity of the color by passing the light, that is photo means light. Okay. So, we are going to pass the light to the solution, and thereby we are going to measure the intensity of the color, and we are going to write what and all the values we have got. So, that is what is called a spectrophotometry. Okay. So, in this spectrophotometry, by measuring the intensity of the color in terms of percentage transmission or absorption or optical density, so we are going to measure the quantity of the substance. Okay. So, in the same way conductometry. So, as I said, conductometry is an instrument which is used to measure the conductance. Okay. So, conductance is the reciprocal of resistance. So, resistance means each and every solution will have resistance towards electricity. It will not allow the electricity to pass through it easily, but certain solutions or certain substances which can dissociate into ions, so that is cations and anions in a very precise manner. So, those ions, that is cations and anions, they are capable of carrying the electricity through it or along with it. Okay. So, that is when an electrodes are inserted to that solution. Okay. So, when a cathode and anode is inserted to the solution of the compound, which can dissociate clearly into cations and anions. So, their anions will be attracted towards the cathode, and cations will be attracted towards the anodes. Okay. So, this is the common phenomenon what it is going to happen when the electrodes are inserted into a beaker of beaker containing a electrolytic solution. Okay. So, conductometry is an instrument which measure the conductance of the substance. Okay. So, it is going to measure the conductance of the substance. Okay. So, as the name itself says, conducto, that is conductance. So, metry means measurement or to write the conductance. So, that is why conductometry instrument is used. And in the same way, potentiometry. So, the potentiometry is an instrument which is used to note down the electromotive force which has been possessed by the ions. Okay. So, the cations or anions. So, they will possess a electromotive force when an electrode is inserted to it. Okay. So, the electromotive force is the potential energy which is going to be exhibited by those particular ions. Okay. So, that will measure the EMF, and the instrument which is used to measure the potential energy of the ions is called as potentiometry. And in the same way, polarography is also used. So, the polarography is used to measure the polarity and also how the ions are going to act in a electrolytic solution. So, that will be measured. And the mass spectrometry, as the name itself says, so mass spectrometry is a technique where we are going to measure the mass of the substance. And flame photometry. So, flame photometry is an instrument which is used to measure the concentration of the alkaline earth metals, okay, alkali and alkaline earth metals. So, we are going to measure the concentration, say for example, the percentage of sodium, potassium or calcium which is present in various different samples like water sample or soil sample or blood fluids or blood plasma. Okay. So, sodium, potassium, calcium is present in our body also, it is there in our body, and also it is present in environment. So, the percentage of alkali and alkaline earth metals will be determined by using flame photometry. That is because these alkali and alkaline earth metals, they are very sensitive towards the flame, and those metals will have characteristic color when they come in contact with the flame. Okay. So, this helps us to determine, so in what concentration does that particular metals are present in that given sample will be determined with the help of flame photometry. Okay. And next spectroscopic methods like nuclear magnetic resonance spectroscopy and electron spin resonance spectroscopy, infrared spectroscopy, visible and UV spectroscopy. So, all these are different instrumental methods which we are going to use for the chemical analysis. Okay. To summarize in briefly, spectroscopic methods like nuclear magnetic resonance spectroscopy, NMR spectroscopy means, so it says us so how many protons are present or how many carbon is present, how many phosphorus or sulfur is present. So, all these the number of protons which has been present in the given organic compound. So, that can be determined with the help of nuclear magnetic resonance spectroscopy. And electron spin resonance spectroscopy, it is also a method of the spectroscopy which is widely used in inorganic chemistry to understand the electron spin. Okay. So, if you see the structure of an atom, so we know that an atom consists of nucleus and the electrons revolve around the nucleus. Okay. So, each electron will revolve in its own axis and therefore, it has got spin. So, each electron will revolve around in its own axis. So, that is why it will exert a spin, and also while revolving around the nucleus it will form orbitals. Okay. So, that orbital will also form orbital angular momentum energy. Okay. So, all these will be studied in electron spin resonance spectroscopy. And next is infrared spectroscopy. So, infrared spectroscopy it helps us to determine so what functional group is present by studying the vibrational pattern between the two atoms which are present in the compound. By studying the vibrational and vibrational arrangement and the force of attraction between the two atoms. So, we can determine the functional group which is present, to which class of organic or to which class does that particular compound belongs to. All these informations will be get by performing the infrared spectroscopic instrumental method of analysis. And for the visible and UV spectroscopy, so this visible and UV spectroscopy mainly it gives us so what sort of bonding, whether it has a sigma bond or pi bond. So, whether the chemical bonding between the two atoms or the molecules is made up of double bond or triple bond. So, all these informations can be get by visible and UV spectroscopic technique. Okay. So, these are some of the instruments which we are going to use to study the properties and also in terms of studying the properties like optical density or intensity of the color or the concentration by measuring the concentration of the solution. So, we are going to estimate the quantity of the substance. So, in how much amount does that particular constituent of substance is present. So, all these informations will be gathered or obtained by performing instrumental methods of analysis. Okay. So, this is all about instrumental methods of analysis. So, now we will see what are the advantages of instrumental methods of analysis. So, for this since we are going to do the experiments with the help of instrument, definitely small amount of sample can be used. We need not to use large amount of compounds like 5 gram or 10 gram of compounds is not necessary for instrumental methods of analysis. Very small amount, sometimes it might be in a mg also, mg scale also, that means milligram scale also. For NMR spectroscopy, you believe it or not, so even 5 mg or 10 mg of compound is more than enough to do the NMR spectroscopy or mass spectroscopy. Okay. So, even for spectroscopic technique, so sometimes we will prepare the solutions in mgs. Okay. So, that is 0.1 mg, 0.2 mg, 0.3 mg. So, in that successive increasing in the quantity of the substance, we are going to prepare a series of solutions. Okay. So, that means, so very small amount of sample can be used. So, this is one major advantage of instrumental methods of analysis. And the next advantage is the determinations can be done quickly. Okay. So, all the determinations will be made quickly within fraction of seconds. If the solution is prepared and if the instrumentation is already been calibrated, so we just have to insert that solution which is under examination and switch on the instrument. So, within fraction of seconds we will get the readings. Okay. So, the determinations can be done quickly and more accurate and reliable results can be obtained, okay, than performing in hands by hands. We humans, we will make lot of errors. So, while preparing the solutions we will make errors. So, you would have observed while doing the titrations only. Okay. So, while performing the titrations we will not get concordant values. So, why we will not get concordant values means, so there will be a variation in pipetting out the titrant or while adding the indicator or while adding the titrant from the burette, there will be a mistake because definitely humans will make mistakes, but the instruments, so they will not make mistakes unless until we have prepared the solutions wrong. Okay. So, that is why, so more accurate and reliable results can be obtained in instrumental methods of analysis. And the fourth advantage is highly sensitive instruments are available and measurements can be done in parts per million scale. Okay. So, this is very interesting. So, I was talking about milligram scale, but even in parts per million means in millions, in million concentration, only one part means that might be 1 mg or 1 gram in thousand or millions of scales. Okay. So, parts per million scale also can be determined or can be examined by using highly sensitive instruments. Okay. So, in recent days, so as the technology goes on improving, so we will have highly sensitive instruments. So, by using that highly sensitive instruments, we can measure the unknown concentration or the sample in parts per million scale also. Okay. So, these are the main advantages of instrumental methods of analysis. And next is disadvantages of instrumental methods. So, cost of equipments are high. So, this is common because when it is highly sensitive, when it is an highly sensitive instrument, definitely the cost will be high. Okay. So, the cost of equipments are high and an initial or continuous calibration of the instrument is required. Yeah, this is very important. While using an instrument, so before performing any experiment, we have to calibrate that instrument. Okay. So, the calibration will take some time. The calibration means we have to adjust the wavelength, we have to see whether the readings obtained will be correct. So, we have to check, we have to do it with some sample or a reference solutions. We have to use reference solutions and calculate or calibrate the instrument. Okay, so an initial or continuous calibration of the instrument is required, and specialized training is required to handle the instruments. Okay, so these NMR, mass spectroscopy or IR spectroscopy, all these instruments we cannot handle it. We should take some training. Okay, so only the trained people can use or can operate the instruments. Okay, and sensitivity and accuracy depends upon the instruments. Okay, so if you use highly sensitive instruments, definitely the sensitivity and accuracy will be more, and if it is of low sensitivity, then definitely the results what we are going to get in terms of sensitivity and accuracy will be low. Okay, so these are some of the disadvantages of instrumental methods of analysis. Okay, so now the next topic of interest in this chapter is solvent extraction. What is meant by solvent extraction? So, solvent extraction is the technique for separation mixtures of substances by selective transfer between two immiscible liquid phases. Okay. So, here we are going to separate the two different substances which are present in the sample or the solutions by the selective transfer between the two immiscible liquid phases. Okay, so immiscible means so they will not interact with each other. Say for example, if you take water with any of the organic solvents like chloroform or acetonitrile or DCM, dichloromethane or ethyl acetate. So, water is immiscible with the organic solvents. Okay. So, whereas if you mix up with the sodium chloride or sodium hydroxide, so you cannot make the separation. We cannot say, so this is water and this is sodium chloride. No, we cannot say, because so water and inorganic solvents, that especially inorganic solvents like sodium chloride or sodium hydroxide or potassium chloride, potassium hydroxide, all these are miscible. Whereas, if you take 10 ml of water and 10 ml of chloroform, and if you mix those two solutions, okay, so you can clearly say, so this is water and this is chloroform. So, such type of solvents are called as immiscible solvents. So, there we are going to make out, we can identify two immiscible liquid phases. So, here what happens? The inorganic salts which are present in that particular solution which is under examination, means which has to be separated. So, all the inorganic constituents like sodium or sodium chloride or potassium chloride or whatever the inorganic salts which are present in that particular solution, they will get extracted towards water, because water will make it separate. Water dissolves that sodium chloride or the salts of inorganic salts which are present there, and the organic compound which is present in that solution, that will be extracted towards the chloroform. In this way we are going to separate the two different constituents which are present. So, this is what is called as solvent extraction. I repeat, solvent extraction is the technique for separation mixtures. Okay, so when we have a mixture of both inorganic salts and organic compound, so this is a mixture. We have a mixture, we have taken it in a beaker. So there we have organic compound, say for example amino acid. So we have amino acid, along with that amino acid we have mixed sodium chloride or potassium chloride. Okay, so in that mixture, when that mixture is extracted to the chloroform, to that, so there the amino acid will get dissolved in chloroform, and the sodium chloride or potassium chloride which is present, so that will get dissolved in the water. So since this is a immiscible liquid, So, chloroform and water is the immiscible liquid. So, we will have a separation between the two solutions. We can clearly identify the two phases, that is, one is water phase and another one is organic solvent phase, that is chloroform phase. So, we can separate it. How we are going to separate? By using separating funnel. We have instrument, it is a glassware which is called as a separating funnel. So, that you can see it once you come to the classes, you can see it, or I will show that in Google class. So, how a separating funnel looks like. Okay. So, when we take this solution in the separating funnel, depending upon the polarity, okay. So, chloroform has got higher density and it has got less polar compared to the water. So, it will be situated in the down part of the separating funnel, and water will be floating on the layer of chloroform. Okay. So, we can clearly separate it. So, in this way, so solvent extraction is the technique for separation mixtures of substances by selective transfer between two immiscible liquid phases. So, here most of the solvent extraction procedures are based on the extraction of non-polar uncharged species from an aqueous solution into an immiscible organic solvent, or the extraction of polar or ionized species into an aqueous solution from an organic solvent. Okay. So, in this way, so the solvent extraction is carried out. So, the solvent extraction or liquid extraction is considered to be most popular and versatile technique for separation. So, this is because so it does not require any sophisticated apparatus or instruments except a separating funnel. Okay. So, just what we need is we need a separating funnel and a stand which is used to keep the separating funnel, a separating funnel and a separating funnel stand. So, if we have that, so then definitely so we can do the solvent extraction. So, that is why so this it has become most popular and versatile technique of separation. And next the separation can be carried out in macro scales as well as in micro level. Okay. So, we can do the separation in macro scale level and also in micro scale level. Okay. So, due to these two reasons the solvent extraction or liquid extraction is considered to be most popular and versatile technique of separation. Okay. So, I am going to stop it here. So, in my next class we will see the basic principle of solvent extraction and what are the applications of solvent extraction. So, thank you dear students.